Welcome back to the Open HBI lecture Code of Life. This lecture focuses on the real-world use case, oncology, because oncology is very prominent and very important, as there is not a kind of standard therapy where you can follow and find the best treatment for one patient. You need to investigate very important patient specifics. First of all, I'd like to introduce you the actors in oncology. Then I want to give you an understanding of some specific terms used in oncology. And later on, I want to sum up and show you how you can use the information about genetic code, the DNA, in this context, in this really practical context. First of all, which are the actors? So I tried to bring here only three parties to the table. The first one is, of course, the patient. He was diagnosed with some severe cancer, and he is looking, of course, for the best treatment in a really short period of time. Typically, cancer disease is challenging because these cancer cells are growing and growing without any notice, and once you notice it and you have some problems with that, it's typically at a late stage, and therefore if you have some challenge to find a cancer, it really time matters to keep it short. The second party here in that game is the clinicians, of course, and the clinicians aim for the treatment of the patients. They try to find the best therapy, and of course the best therapy with the least amount of side effects or any kind of severe effects. So they try to investigate the patient, learn about the patient specifics, and understand how the patient could benefit from a certain therapy. And last but not least, the researchers. The researchers here are important because they back up the clinicians. They produce a lot of laboratory work, produce data, and they find new insights in that context. So therefore, these three parties are somehow connected at every time. And we will focus now in this lecture, of course, on the clinicians because they want to find the best treatment. And what they want to do is they want to do informed decision making. Informed decision making means they want to involve as much as possible information about a patient, a specific patient, to find the best therapy for one individual. So what they want to find is the best available treatment option. They want to investigate a lot of diagnostic data because today it's not a challenge of creating data. You can create huge amounts of data in a really short period of time. For instance, sequencing today is not like a 10-year time frame. You can sequence the whole genome of one patient within some hours, less than a day. And this is really just 10 years after the Human Genome Project discovered the whole human genome. And last but not least, they want to be able to exchange these informations. They want to exchange information, of course, with the patients to inform them about available treatment options, but also with other clinicians, other oncologists, other experts to understand and explore alternative ways of treatment. So what is our vision we want to share? The vision is an interdisciplinary tumor board. Today, each cancer case is presented in a at least clinic-wide tumor board where a lot of clinicians are at a table and discuss a certain patient to understand what is the proposed therapy by the clinician and what are alternatives and what could be side effects. This interdisciplinary tumor board goes a little bit further. It says first, we do not have to limit the knowledge to one clinic. We can incorporate experts from all over the globe. You see here some, on the right-hand side, some people from Shanghai. Some people are connected via video conference. They participate in a virtual tumor board. On the other side, the participants here in the tumor board not only oncologists and clinicians. You see here also bioinformaticians, you see genetists, and a lot of more parties are involved, most probably also software engineers in future, to do statistical analysis to understand where are the data and to bring them together in that moment where the discussion takes place. So you do not have to reschedule a next tumor board for one patient. You can access all the data you have at hand in that very important moment. And here you see the requirement also to have this kind of analysis and this kind of data acquisition really available in real time. You need to have access to the data. You need to have to combine the data in that moment when the experts are talking about that. So first of all, how to come to this vision so that the doctor you see here on the right-hand side is able to find the best treatment and initiate the best treatment from the first point in time. We need to introduce some terms because they are commonly mixed in that field, and therefore I'd like to distinguish them here. First of all, I'd like to introduce stratified medicine to you. So the term stratified medicine is actually mostly in the European Union present. And it refers to the identification of subgroup of patients. And that this kind of subgroups or cohorts are important because you can find certain kind of indications where you can put people into cohorts, and these cohorts are treated in a very similar way, right? Stratified medicine is a kind of existing way where you want to find hopefully really small groups, because if you have one big group, like a large group like all diabetic patients, it's too large. You want to have a subtype of diabetic patients, male, the age is higher than 50, and they have this and this preconditions. Personalized medicine, in contrast, goes a step further by using even targeted medicines for specific changes, for instance, in the DNA, and they also involve the patient's genotype. The second term, as we were talking about personalized medicine, is personalized medicine. Personalized medicine is the concept that selection of a treatment should be tailored according to the individual patient's specific characteristics. So what does it mean? Of course, clinicians and medical experts were always working like this. They always treated one patient at a time. However, here it says get most patient-specific characteristics out of the patient's anamnesis, out of the history of the patient, put them together and use them as kind of decision-making. And you see, in contrast, is that standard of care deciding, because there you take a lot of people treated in a similar way and say the average of these people were treated best like this and this. So personalized medicine is really a more fine-grained, tailored approach in that field. And last but not least, and this is the reason why also President Obama is mentioned here, this is precision medicine. And he was introducing the Precision Medicine Initiative just 1 year ago in 2015, and which is a big scientific program in the United States and worldwide. And the precision medicine is a little bit different term, but it's very similar to the personalized medicine approach. Precision medicine is an emerging approach for disease treatment and prevention. This is important, prevention, that takes into account individual variability in genes, environment, and lifestyle for each person. And you see, it's even more. It's a more and broader focus. It says, I'd like to introduce not only the treatment of persons which are currently sick, I want to prevent them from getting sick. And therefore, I'd like to measure as most information and data which is important for any kind of indication, early indication. This is the difference between precision medicine and personalized medicine. So let's come to a concrete example as we are here in the oncology world. I'd like to introduce you a so-called case. And this case is characterized by some metadata. For instance, we have a patient, let's call her Jane Doe. She's 48 years old, female, never smoked, and lived always in a smoke-free environment. However, she was diagnosed just recently with a non-small cell lung cancer diagnosis. And you see, non-small cell lung cancer for you it's a lung cancer case. This is typically indicated by this kind of radiology picture. This is an example because at that late stage you would see even more of these shadow areas. However, the question is, typically this is associated, this kind of disease is typically associated to smoking, right? And how did she get this kind of disease? So therefore what happened, they sent the tumor material, which was selected by a biopsy, to a laboratory to analyze this. And typically what you would do is you would need someone, Dr. House, to solve all the issues because he knows basically the best answer. But what is done today in the clinical environment? And therefore I'd like to introduce the process to you as a kind of simplified view on the things. First of all, what you see here on the top is the clinical department. This is where the clinician has contact to the patient. And the first thing is the tumor will be biopsied, so you get a sample of the tumor material and maybe also a sample of the healthy tissue. And these two samples were sent to the wet lab department. The wet lab department could be part of the pathology department. And in the wet lab department, they do all the preparation work which is necessary to process these samples in a laboratory environment and to prepare them for the actual sequencing. And the third party here is the sequencing, which results in a lot of data. And this is done in the laboratory. And in that moment when we have data, we can come to the next slide. This is the bioinformatician department, where they have to reconstruct the whole genome of the patient. They have to identify the variants. And this is done by computer scientists, and in the end you will have a list of variants, and this is what you see here, down here. This list of variants might be pretty large. Typically what you should know is typically human by human differentiates by about 1% in the human genome. If you consider 3.2 billion base pairs in the human genome, you know, you have even millions of variants where we are different from each other. But all of these variants make us somehow individual. Not each of these variants is necessarily a kind of trigger for this kind of disease. So you need to figure out which kind of variant is really responsible for the oncology use case here for the lung cancer disease. And which one is maybe only the hair color, dark versus light, or the eye color or something like this. So therefore you have to process each of these variants, these of million variants. And then you have to understand for each of the variants which genes are involved, what is the gene function, how is this involved in a cellular pathway, maybe which kind of products are created within the cells. And you have to understand what kind of therapies are available by reading publications, by finding similar cases, going through clinical trials and so on. I don't want to go too much into detail here, but you see it's a lot of search and research work here which is conducted, and this takes a lot of time. And last but not least, you get a kind of assembled report. And this report is really short. It's 1 to 3 pages long, because this assembled report is sent back to the clinician, and the clinician now can use this kind of report to decide what kind of therapy is the best. Of course, maybe they can start with radiation therapy, or they can start with a kind of chemotherapy, or they even start with a clinical trial which is exactly targeting these changes in the genes. So if you find out in that report that there are certain kind of genetic variations, genetic mutations are responsible for this tumor disease, you can try to address them explicitly. And then, of course, you start the therapy, and even if this is not, even if this appears to be a long process, this needs to be cut down to some hours, to some days to start with the best therapy, because these tumor cells still continue to grow in that moment, right? So what you have learned today is you know what is the important thing in the precision medicine, personalized medicine, and stratified medicine approaches. They are very similar, but they are different in the specific terms. You have learned about the oncology use case, the lung cancer use case, and you see how complex this clinical process is, and you see this is just a simplified process. And what you learn next time is what happens with the biopsy? How can we get to the information, exactly what was done in the bioinformatician department? How can we get the important information out of the biopsy sample? What are the cell components? How do we extract the DNA? And how to build then this kind of report for the clinicians. So thank you very much for attending today. I'm looking forward to see you next time.